Bacteria. They are everywhere. On your skin, in the food you eat and the water you drink, even in the air you breathe. Bacteria seem like elusive organisms to most people, something that we cannot see with our eyes alone, causing devastating diseases such as cholera, the Black Death, or just common food poisoning. Yet there are some bacteria that are beneficial to us by protecting you and I from some of the same pathogenic microorganisms causing these diseases. They are utilized by scientists to push the boundaries of science, production of pharmaceutical drugs, and much more. Let's talk about bacteria. Look at your hands. They're crawling with bacteria. But don't worry, they're good bacteria. Bacteria living on your skin or your gut is what is known as commensal microorganisms, a relationship that is mutually beneficial to us and the bacteria. Bacteria in your gut aid in digestion by breaking down compounds that your body cannot, for example, fiber. Fiber is a collection of complex carbohydrates that are derived mainly from plants, however cannot be completely broken down by human digestive enzymes. This is where commensal gut bacteria come in. They possess different enzymes that can break down complex carbohydrates into simple sugars. This process can contribute as much as 10% of the calories that our cells require. Bacteria that colonize your gut not only aid in digestion, but also provides us with a biological line of defense. They directly inhibit the colonization by pathogenic and opportunistic microorganisms by competing with them for nutrients and fully occupying the niche. They can also prevent pathogenic microorganisms from invading the epithelial cell linings of our guts by competing or blocking the pathogenic microorganisms from gaining access to receptors required for their entry. Biological protection doesn't only occur in the gut, but also occurs in our lungs as well as on our skin. Of course, some bacteria can bypass this line of defense, resulting in infections. One such infection is cholera, which is a disease that is widespread in poorer countries that do not have access to safe drinking water and have poor sanitation. The causative agent is the bacterium called Vibrio cholerae. Symptoms include diarrhea and vomiting, resulting in dehydration and death if treatments are not administered quickly enough. Vibrio cholerae spreads through the fecal-oral pathway by drinking water that has been in contact with feces from an infected individual, or by eating food prepared by an individual with fecal residue on their hands as a result of poor sanitary practices. Vibrio cholerae, when consumed through food or water, reaches the stomach, where a majority of the cells die due to the stomach acid. However, few can survive reaching the intestine. While in the intestine, they penetrate the mucous lining of the epithelial cells and adhere and colonize the microvilli where they replicate. They express and secrete different virulence factors and toxins, for example, the cholera toxin. Cholera toxin is internalized by different cells and subsequently activated, which binds to the human protein partner ADP ribosylation factor 6, catalyzing it to increase adenylate cyclase activity. This results in increased intracellular cyclic AMP concentrations, allowing the overactivation of protein kinase A. Active protein kinase A phosphorylates chloride channels, leading to an efflux of chloride ions into the intestinal tract. This causes water and other ions to follow, resulting in diarrhea. Vibrio cholerae is shed through fecal matter and passed on to the next victim through ingestion. Outbreaks can be widespread, with 7 of the largest outbreaks of cholera in the last 200 years resulting in millions of deaths. As a result, there is a push for greater access to clean drinking water and education about the importance of sanitary practices in poorer nations to prevent the next cholera outbreak. Bacteria are not only a problem in poor nations, they can be lurking in your kitchen and strike if you're not careful. If you prepare and eat chicken, you will probably know the importance of fully cooking the chicken and washing your hands, the knife and the cutting board that has been in contact with raw chicken. This is because raw chicken can be contaminated with a genera of bacteria such as Salmonella, which result in gastroenteritis, an infection of the digestive tract. Salmonella is a gram-negative bacteria, meaning that it consists of a thin layer of peptidoglycan surrounded by an outer membrane. The outer membrane consists of lipopolysaccharide, or LPS, which is an endotoxin. Once ingested, the stomach acid kills a majority of the Salmonella cells. However, from the dead cells, these LPS molecules can be released from the outer membrane and be uptaken by nearby epithelial cells. The endotoxin binds to specific receptors on phagocytic cells, which are immune cells that eat foreign material. Once bound, it promotes the release of pro-inflammatory molecules like cytokines, resulting in localized swelling and inflammation, a common symptom of gastroenteritis. The symptoms of food poisoning are not usually severe and do not last a long time, so most people don't require medical help. Not all bacterial infections have non-severe symptoms. One of the most devastating outbreaks caused by bacteria in human history is the Black Death in the 14th century, which resulted in an estimated 20 to 200 million deaths. The causative agent is Yersinia pestis, a gram-negative rod-shaped bacterium spread to humans through rat fleas. Flea bites allow the bacterium to enter by passing our first physical line of defense, the skin. Once inside the body, some are detected and eaten by white blood cells, for example, neutrophils and macrophages. Undetected bacteria migrate to the lymph nodes, escaping phagocytosis and allowed to replicate, resulting in swelling of the infected lymph nodes. Yersinia pestis possesses a type 3 secretion system. Similar to a syringe, it contains a needle-like structure called an injectosome, which can directly penetrate its host cell and inject proteins such as virulence factors to aid in infection. Some virulence factors injected by Yersinia pestis are called Yersinia outer proteins, or YOPs, linked to immune cell evasion. Remember the Yersinia pestis cell that was eaten by the macrophage? It has injected a virulence factor called YOPJ, which is an acetyltransferase, an enzyme that transfers an acetyl group. YOPJ binds to kinases important for different cell signaling pathways and transfers these acetyl groups onto key amino acids. This disrupts cell signaling and results in apoptosis, where the macrophage is targeted for cell death. This allows the bacterium to escape destruction by phagocytosis and allowed to replicate. Yersinia pestis today is no longer a large issue, with antibiotic treatments such as streptomycin and tetracycline able to effectively kill Yersinia pestis. Antibiotics have been crucial in our fight against pathogenic bacterial infections. However, overuse and improper use has prompted the emergence of antibiotic resistance. Bacteria that have gained antibiotic resistance have gained a circular piece of DNA known as a plasmid, which has the antibiotic resistance gene encoded on it. Plasmids can be readily lost by the bacteria to the environment and gained by other bacteria of the same or different species. This is known as horizontal gene transfer. Improper use of antibiotics, such as using it to treat viral infections or not finishing a prescription, results in the emergence of antibiotic resistance. Taking an antibiotic pill kills most but not all bacteria. Bacteria with antibiotic resistance are not readily killed by that one antibiotic pill and often takes a higher and sustained dosage to be killed. As such, a higher proportion of bacteria that have survived will contain the antibiotic resistance plasmid. As the bacteria replicates, those that have picked up the antibiotic resistance plasmid will survive and in turn replicate as well. There is a selection pressure for the bacteria to have this antibiotic resistance plasmid, and as a result, the emergence of an antibiotic resistant strain of bacteria. Some of these strains can be resistant to multiple antibiotics. For example, methicillin-resistant Staphylococcus aureus, which is resistant to multiple groups of broad-spectrum antibiotics and arose through the horizontal transfer of genes as well as natural selection. Methicillin-resistant Staphylococcus aureus is very common in hospitals where a high proportion of the patients are elderly, immunocompromised, or susceptible to the infection. With multiple antibiotic-resistant bacteria becoming more of a problem, there has been a push for research into alternative antibacterial treatments, such as antimicrobial nanoparticles, bacteriophage therapies, and natural alternatives. Don't worry though, this doesn't involve research in injecting disinfectants. Sorry, Trump. Scientists have taken advantage of bacteria's ability to horizontally transfer these genes, such as antibiotic resistance. In these plasmids, we can insert a particular gene that we want the bacteria to translate into proteins. To ensure that we are only growing bacteria that contain the plasmid with the gene that encodes for the protein that we want, we also insert an antibiotic resistance gene in the plasmid. In the media that we use to grow the bacteria, we supplement that with an antibiotic corresponding to which antibiotic resistance gene we have inserted, for example, ampicillin. For any bacteria that is growing in this media, there will be a selection pressure for the bacteria to contain antibiotic resistance. This prevents the contamination of unwanted bacteria in the growing media, and also selects against the bacteria that have lost antibiotic resistance plasmid and conversely can no longer produce the particular gene of interest. This selection process allows scientists to mass synthesize different proteins in bacteria for pharmaceutical use, for example, insulin. Bacteria resemble the commensal bacteria that aid us in everyday processes such as digestion and a biological line of protection. The bad, the causative agents of several infections and outbreaks responsible for millions of deaths, as well as the ugly, the multiple antibiotic-resistant bacteria that arise through the improper use of antibiotics. With such a diversity of bacteria in the world, there is much more we can learn about the processes they perform and the mechanisms of the epidemiology they can cause. All the information that we uncover is beneficial to us as we push the boundaries of science that one bit further.